Aloha. I will now continue with my presentation of the second part of Chapter 15. Last time we discussed the regulation of glycolysis by regulating the hexokinase enzyme. Today I will focus on the regulation of glycolysis and gluconeogenesis by regulating PFK1 and FBPase1 enzymes, and also the regulation of pyruvate kinase and pyruvate carboxylase. There is a balance between the activity of these enzymes, and there are multiple factors that influence their activity. So the regulation is quite complex, but it still makes sense. Alright, so something interesting about the PFK1 enzyme is that it uses ATP as a substrate. It catalyzes the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. And it uses ATP in that reaction. So ATP is a substrate, but at the same time, at high concentration of ATP, the enzyme is inhibited. How is this possible? The substrate would activate and at the same time, under different conditions or at higher concentration, deactivate or inhibit the enzyme. Why is that the case? If you remember, we talked about this, but for a different enzyme. But the same rule here is true for PFK1. So why is that? How is this possible? Well, there are two binding sites for ATP. One binds ATP as a substrate, and the other one, the allosteric site, binds ATP but only when ATP is at higher concentration. And when ATP binds at that site, it inhibits the enzyme. The next question I would ask is, which of these sites do you suppose has higher affinity for ATP? The substrate site or the allosteric site? Think about it. The substrate site uses ATP to push the reaction forward. The allosteric site would bind ATP only at higher concentration of ATP so that it will inhibit the enzyme. So which of these two sites will have higher affinity for ATP? Of course, the substrate. Now, let's look at this graph. Which of these two curves represents the activity of PFK1 at high ATP concentration, and which of the two represents the activity of PFK1 at low concentration of ATP? Again, what happens with the activity of PFK1 when we have high ATP concentration? It inhibits it. That means that the lower graph, the graph in magenta or dark red color, represents the activity of PFK1 at high ATP concentration. Alright. Now, how would the citrate, ADP and AMP affect PFK1 and FBPase1 enzymes? Well, these figures show you that ATP would inhibit the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate because we have plenty of energy and we don't want to stimulate glycolysis, while AMP and ADP would activate it in order to produce fructose 1,6-bisphosphate that will stimulate glycolysis. The same for citrate. Citrate is, you don't know that yet, or perhaps you've forgotten from general biology, but citrate is a downstream product of pyruvate. Actually, it's the first product of the citric acid cycle. So if we have higher citrate concentration, that means that we have plenty of ATP, that the TCA cycle is somewhat inhibited, and we have plenty of citrate, that means that there is no need for glucose to be degraded to pyruvate and for pyruvate to become acetyl-CoA. So citrate will inhibit glycolysis. Now, here we also see on this figure that there is another metabolite called fructose 2,6-bisphosphate that plays a role in the activity of PFK1 and FBPase1. So let's see, well, let me actually backtrack a little bit and leave fructose 2,6-bisphosphate for a moment, and I'll come back to it in a moment, but I just wanted to focus again on ATP and ADP. Alright, so you can see on figure 15.15 that high AMP stimulates glycolysis, ADP as well, and inhibits gluconeogenesis. Right? Yet, but on figure 15.6, the figure shows that, for example, in the heart, glucose uptake is stimulated, glycolysis is stimulated as well. That's at low ATP but high AMP. The same thing happens in the muscles. Glucose uptake is also increased. In the liver, however, we know that gluconeogenesis is activated while glycolysis is inhibited. So how can high AMP inhibit and stimulate gluconeogenesis at the same time? Or inhibits and activates glycolysis at the same time? Isn't that confusing? The real answer is that it can happen, well, that can happen if the reaction occurs in two different places under two different conditions. So in this case, the seemingly dual response is based on the type of tissue. In the liver, gluconeogenesis is activated. In the heart and the muscle and other tissues is inhibited. In those tissues, glycolysis seems to be activated, to a point though, because really there is a shift towards the use of fat for energy. But in the liver, glycolysis is fully deactivated. So we can see, well, in addition to that, we already know that the liver is not dependent for its ATP to be produced from glycolysis, but instead it depends primarily on fatty acid degradation. Now, when fatty acids are degraded, they produce massive amounts of acetyl-CoA, which is, you don't know that yet, but it's a product of the fatty acid degradation. And that acetyl-CoA is used by the citric acid cycle to produce ATP. However, when acetyl-CoA is increased in concentration, that acetyl-CoA stimulates gluconeogenesis by stimulating pyruvate carboxylase, which is the first enzyme of gluconeogenesis. We're going to get to that at our last slide in today's presentation. But I'm mentioning it here so that you can make connections. So the liver is the only tissue which can raise blood glucose levels. The muscles have glycogen deposits, but they only serve the muscle. The liver is the one that uses its glycogen deposits and gluconeogenesis to support the brain and the entire body with glucose. So it is very important that in the liver gluconeogenesis is activated while glycolysis is inhibited. While all the other tissues, glycolysis, if anything, is activated. So we can see that at the same time, in different tissues, we have a variant level of regulation of the same process. So you can see that the answer is not always very simple when we talk about biochemistry and the chemistry of life in general, or the regulation of these processes. A lot of times, in my presentations or in a textbook, we try to make things clear, and we separate independent processes or reactions so that we can clarify and make sure that when we study, we actually understand the main purpose or the main function of an enzyme or a reaction or a pathway. But the reality is that the regulation and the communication between these pathways or cells or tissues is a lot more complex. And sometimes it's not very easy to simplify things. You can see it here. AMP inhibits gluconeogenesis and the rest of the tissues, but at the same time activates gluconeogenesis in the liver. So how does that happen? How do we do that? Hopefully that will become clear by the end of today's discussion. Now, let's focus now back to fructose 2,6-bisphosphate. Alright, this is a metabolite that is produced by one protein, but that protein has dual function. That enzyme has dual function. If the enzyme is phosphorylated, it acts as FBPase2 that degrades fructose 2,6-bisphosphate to fructose 6-phosphate. If the enzyme is dephosphorylated, the enzyme acts as PFK2, which converts fructose 6-phosphate to fructose 2,6-bisphosphate. Alright, so let's look at now these graphs and try to make sense, or try to see how these graphs actually agree with this graph, showing that fructose 2,6-bisphosphate activates glycolysis and deactivates gluconeogenesis. So how does the concentration of fructose 2,6-bisphosphate affect the function of PFK1 and FBPase1 enzymes? Let's look at this graph. Activity of PFK1 based on fructose 6-phosphate concentration, which is the substrate of PFK1. In the presence of fructose 2,6-bisphosphate, we have this activity. In the absence of fructose 2,6-bisphosphate, the activity seems almost gone. You have to have very high concentrations of fructose 2,6-bisphosphate in order for PFK1 to function, even a little bit. So it seems that the presence of 2,6-bisphosphate is required for the activity of PFK1, meaning that the presence of fructose 2,6-bisphosphate activates or supports glycolysis. Now let's look at the other graph. This is FBPase1 activity versus, well, with respect to increased concentration of its substrate, which is fructose 1,6-bisphosphate. In the absence of fructose 2,6-bisphosphate, we see a very steep activation of FBPase1 with increase of fructose 1,6-bisphosphate. In the presence, we see severe inhibition of FBPase1 activity. So, based on this graph, we can make the conclusion that fructose 2,6-bisphosphate inhibits the function of FBPase1, meaning that it inhibits gluconeogenesis. As a matter of fact, fructose 2,6-bisphosphate acts almost as a coenzyme for PFK1, because in the absence of it, PFK1 is virtually inactive. Alright, now the production of fructose 2,6-bisphosphate is hormonally regulated. In the liver, very important that it's in the liver, glucagon inhibits glycolysis and stimulates glucose production from glycogen and gluconeogenesis. Again, why? To support normal blood glucose levels for the brain and all the other tissues. Glucagon is released when we have low blood glucose level. I always connect it with when glucose is gone, then glucagon is released to help keep the glucose levels normal. Insulin, in general, inhibits gluconeogenesis and stimulates glycolysis and glycogen synthesis. When is insulin secreted by the pancreas? When we eat, when we have high blood glucose levels. The function of insulin is to push glucose and other metabolites into the cell for degradation or for storage. Alright, so these are the functions of these two hormones with respect to glycolysis and gluconeogenesis. So what effect would these two hormones have on the activity of the enzyme catalyzing the fructose 6-phosphate conversion to fructose 2,6-bisphosphate, or the reverse process? Which hormone would lead to phosphorylation of the enzyme? Alright, so this is an excellent mind game. Think about it. What does phosphorylation of the enzyme responsible for the production or the degradation of fructose 2,6-bisphosphate would do? If we phosphorylate the enzyme, we activate FPPase2, which will decrease fructose 2,6-bisphosphate. So, which of those two enzymes would try to stimulate gluconeogenesis by lowering fructose 2,6-bisphosphate and turn off glycolysis, again because it would lower fructose 2,6-bisphosphate? Obviously, glucagon. Glucagon would phosphorylate the enzyme, thus it will decrease fructose 2,6-bisphosphate production that will shut down glycolysis and stimulate gluconeogenesis. So which hormone would lead to an increase of fructose 2,6-bisphosphate? The opposite, the insulin. The insulin will dephosphorylate the enzyme, leading to increase of fructose 2,6-bisphosphate, thus stimulating glycolysis. Alright, so whenever we talk about these processes, I know this can get really confusing. So take it slow. Make a pause. Draw it if you must on a piece of paper. Try to figure it out, and don't try to memorize. Try to think logically. Hopefully with how I'm approaching these questions, I'm trying to give you also an example how to think about them. Just use logic. Try to figure out the answer rather than memorize the answer. Alright, so what I just said is basically described in this figure. I really love figures because they really show everything graphically, and it's easy to comprehend. So again, glucagon through a cyclic AMP-dependent protein kinase phosphorylates the protein, thus turning FPPase2 activity on. That will degrade fructose 2,6-bisphosphate, thus inhibit glycolysis and stimulate gluconeogenesis. Alright, I just have an interesting question here for you. If I'm telling you that the glucagon will use cyclic AMP-dependent protein kinase, or pKa, what type of receptor would that be? Think about biosignaling 481. If you thought of G protein-coupled receptors, that will be the right answer. Because you remember, G protein-coupled receptors signal through pKa, or cyclic AMP-dependent. Kinase cyclic AMP is a second messenger that gets produced by adenylyl cyclase, which gets activated by G protein alpha. Alright, just a little review and a connection with what we've learned before. Insulin, on the other hand, through a phosphoprotein phosphatase, dephosphorylates the protein, turning on a PFK2 site active that will increase fructose 2,6-bisphosphate, thus stimulating glycolysis and inhibiting gluconeogenesis. Alright, so as if it's not already quite complicated, now I'm also going to add another regulator of this whole process, and that regulator also controls or regulates fat metabolism, and that is xylulose 5-phosphate. This metabolite has the property, well, it's produced in the pentose phosphate pathway from ribulose 5-phosphate during the non-oxidative phase. If you go back to the pentose phosphate pathway, you will see it, that it's one of the first products of the non-oxidative phase of the pentose phosphate pathway that is used in the conversion of ribulose 5-phosphate to glucose 6-phosphate. Now, xylulose 5-phosphates activates fructose 2,6-bisphosphate production by activating a phosphatase enzyme that phosphorylates the enzyme that controls the production and the degradation of this metabolite. By dephosphorylating the protein, it'll turn PFK2 activity that will make fructose 2,6-bisphosphate. What would that do to glycolysis? Activate it. Alright, so what will be the effect of high glucose on carbohydrate and fat metabolism? So high glucose leads to increased glycolysis, leading eventually to the production of acetyl-CoA. Acetyl-CoA is the first product of pyruvate processing towards degradation and energy. So acetyl-CoA, however, is also a precursor for fatty acid synthesis. In other words, when we have too much acetyl-CoA that is no longer needed for energy, it will be used to make fatty acids and be saved as fat. The increased acetyl-CoA, when it's in excess, will, like I said, stimulate fatty acid production. But what does fatty acid production requires? NADPH. Now, when the demand for NADPH increases, and NADPH is getting used, what would that do to the concentration of NADP+? It would increase. And what does that do to the pentose phosphate pathway? It turns it on. It activates it. So more of the glucose 6-phosphate will enter into the pentose phosphate pathway. The pathway generates NADPH, which is used for fatty acid synthesis, and it also generates ribulose 5-phosphate. Ribulose 5-phosphate, as you know, can be converted to ribulose 5-phosphates, which can be used for nucleotide synthesis. But if there's no need of newly synthesized nucleotides, what would happen with that ribulose 5-phosphate? It will be recycled, as you remember, in the non-oxidative phase. To enter the non-oxidative phase, the ribulose 5-phosphate will be converted to xylulose 5-phosphates. Xylulose 5-phosphates stimulates the phosphatase enzyme that dephosphorylates the enzymes that control the production of fructose 2,6-bisphosphate, meaning that will stimulate PFK2 function of that enzyme that will increase fructose 2,6-bisphosphate, which in turn will stimulate PFK1 or glycolysis. So you can see how these are connected. Glycolysis is stimulated, and at the same time fatty acid production is stimulated. Do you see now the connection between sugar intake and fat production? Do you see how, and I will make that point in the future again, how you don't have to consume a molecule of fatty acids, but you can make them just as well yourself, because the best way to save energy in the body is to convert it into fat. When you have an excess consumption of carbohydrates or glucose, the body supports, the metabolism supports the storage of that energy, the storage of that glucose in the form of fatty acids by converting it to acetyl-CoA and to the pentose phosphate pathway provides the necessary NADPH that is needed for the production of fatty acids. All right, now we'll shift gears, and for the remaining of my presentation, I will focus on the pyruvate kinase, which is the last enzyme of glycolysis. Someone might say, well, isn't it too late to, for glucose to be saved? This is the end of the pathway. Well, that may be the case, but you also have to understand that this enzyme is in the balance, or is balanced, its activity is balanced with the activity of the enzymes that control gluconeogenesis. And even if that is the case, that it's the last enzyme, it still offers support or regulatory mechanism for glycolysis regulation. Alright, so pyruvate kinase has two forms, liver form and muscle form, that have somewhat of a different regulation. So as ATP, acetyl-CoA, and fatty acids, you can see it all here, allosterically inhibit both forms of pyruvate kinase. Now, if I were you right now, I would say, does that make sense? And the answer should be, of course it does. All right, ATP, high ATP. Well, yes, it will try to stop the degradation of glucose because we have plenty of ATP, so it will have an inhibitory role. Acetyl-CoA is the product of conversion of pyruvate in its way to the citric acid cycle or in the production of fatty acids. So if you have too much of that, well, of course, it will try to shut down the steps before that. So, of course, it will have a negative effect. How about fatty acids? Well, if you have too many fatty acids, basically the body knows that there's plenty, it will try to shut down glycolysis. And which sometimes that also shuts down the appetite. But I don't think that works that way. So anyway, that's why these products have an inhibitory role, the pyruvate kinase. This is an excellent example of feedback regulation. When the product comes back, binds allosterically, and inhibits upstream reactions in an attempt to regulate itself. Another way to regulate pyruvate kinase is by phosphorylation, which is only in the liver. Now, when we have low blood glucose level, glucagon is released, and through its pKa, or cyclic AMP-dependent kinase, it phosphorylates pyruvate kinase and deactivates it. By deactivating it, it shuts down glycolysis. Now, there are other mechanisms that are in balance with this. For example, even if we have low blood glucose, if the body is under duress or under stress, then we have another hormone that is released, and that is adrenaline. Adrenaline activates pyruvate kinase. Adrenaline activates glycolysis. Why? Because adrenaline is the hormone of fight or flight. The adrenaline's purpose is to increase blood glucose level to have fast and quick access to energy for the body, muscles, brain. The body is under alert. Even though we may have conditions of low blood glucose or decreased ATP in the body, adrenaline will try to stimulate whatever it can for the body to produce as much as possible energy to be able to preserve itself. And that's why sometimes perhaps you feel that shaking, because the adrenaline is released and is trying to increase to a point blood glucose level and the availability for it to be degraded for energy. So again, I probably do this a heavy disservice by sharing all these things, because the regulation is far more complex, and the networking between all these different enzymes, all the different hormones, the different tissues, it's mind-boggling, and at the same time highly interesting. And it's incredible to be able to comprehend how all these reactions work together. Every time I teach this section, I'm reminded of an experience I had in medical school when, in our last biochemistry lecture, the professor drew a map of what happens with the glucose metabolism when there is diabetes in the cell, or when the body or the tissues or the person has diabetes. And I don't remember what exactly he wrote on the board, but I remember this schematic with different arrows, and I remember being amazed and so excited to be able to see that. I think that was probably the only time when I imagined that I might actually become a biochemist, and I would love to do that. Because I was in medical school, of course, my goal was to become a physician, but at that time, I actually realized the beauty of of having that understanding of the normal versus the pathological, and from there to be able to draw conclusions and understanding of how disease happen and how they operate, and then from there, how can we control them and treat them, etc., etc. All right, the last thing I wanted to focus is something that I already told you, and that is the regulatory role of acetyl-CoA. Now, pyruvate, which is the last product of glycolysis, or the first substrate of gluconeogenesis, can either be converted to acetyl-CoA that can enter citric acid cycle or fatty acid production, if it's too much of it, or pyruvate can be converted to oxaloacetate and back to glucose. That decision is controlled to a point, of course, by the balance of these two enzymes, pyruvate carboxylase and pyruvate dehydrogenase complex. Now, we haven't talked about pyruvate dehydrogenase complex. That is the enzyme, we'll talk about it a little bit later. That's the enzyme that converts pyruvate to acetyl-CoA. But the acetyl-CoA, through its feedback inhibition, inhibits pyruvate dehydrogenase that stops the conversion of pyruvate to acetyl-CoA. But when would that happen? When we have high acetyl-CoA. When can we have high acetyl-CoA? Well, either when we have too much pyruvate, too much glucose consumed, and there's too much energy, and now acetyl-CoA is going to fatty acid production, or when we have low blood glucose level and the body shifting towards the use of fatty acids for its energy needs. When that happens, when we start degrading fats and fatty acids, the concentration of acetyl-CoA rises quite a bit, because that's the product. We'll talk about that later. Sorry, I'm telling you too many. We'll talk about that later, but it's hard to make these connections without mentioning these other pathways. But when we have high acetyl-CoA because of increased fatty acid degradation, when we have low blood glucose levels, then that acetyl-CoA will try to stop the degradation of pyruvate to acetyl-CoA and stimulate pyruvate carboxylate that will turn it into oxaloacetate, and which will enter and continue actually the gluconeogenesis pathway to become glucose. So that's primarily in the liver. And that works together with all the other mechanisms to be able to stimulate gluconeogenesis when the glucose levels are low, blood glucose levels are low, to stimulate gluconeogenesis and inhibit glycolysis. Because the purpose of the liver is to generate and to keep normal blood glucose levels so that the brain and the rest of the body can have enough of that energy. Now, here's an interesting question which we're going to answer later. What if the blood glucose levels are so low that the body does not have sufficient fuel for the brain? What would happen then? Well, maybe I can give you a little heads up. Let's say when we have hypoglycemia, or too low or very low glucose in the blood. You've heard perhaps of hypoglycemic coma, or sometimes people that experience hypoglycemia, they can faint. That's a defense mechanism. The body is trying to shut down itself with the brain so that it will not need glucose, so that it will preserve itself, and only the basic rudimentary functions of the body continue to exist, such as heart rate, such as heart beating, so that it will keep the body alive. The brain shuts down. That's why people faint, in order to preserve itself. Otherwise, it will be hurt because there is no glucose. But that's only when this happens fast. What if we experience prolonged low blood glucose level? For example, for people that have not consumed any glucose for days. We hear about people that fast 3 days. I know people that have fasted 40 days. No food at all. On purpose. What would happen then? Well, the body naturally will use fat for its energy purposes, but the brain cannot use fat. So what would happen then? How will the brain be fed when there's no glucose, or very low glucose in the body, because there is no food intake? You can only get so much from amino acids from your own proteins. So how will the body function then? That's an excellent question, which I'm going to answer a little bit later when we'll learn more about fatty acid degradation. I feel like I'm finishing this lecture on a cliffhanger, but it's a very interesting correlation, or very interesting collaboration between glucose degradation in production and fatty acid degradation in production and energy production at the same time. All right, with that, I will end my presentation for today. I will continue with the rest of chapter 15 next time. Thank you for your time.